Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on digestion of carbohydrates. Now, as you can see on the screen, I am showing you one figure. Now, where in our body the process of digestion is taking place? So, there are two places where the process of digestion is taking place. First is in our mouth. When we are chewing the food, we know that from salivary gland, saliva mixes with the chewed food, and in the saliva, salivary amylase enzyme is there. So this salivary amylase digests the carbohydrate in the mouth. Then this is the GI tract or the small intestine is shown, and here this one is the lumen. This is the lumen of small intestine. And here the pancreas is shown. And we know the two pancreatic duct, the main one and the accessory one. These two duct of pancreas, they pour the pancreatic juice in the small intestine, that is the duodenum. Now, in the pancreatic juice, pancreatic enzyme is there, and this enzyme is involved in the digestion of carbohydrate. Now, one setup or the arrangement I would like to show you. See, this arrangement we need to understand what is this. So, here, as you can see, the lumen is shown, and this lumen is of small intestine, and this green color cell, this cell, these are the epithelial cell of the small intestine. So around the lumen we know the mucosa is there, and the layer of epithelial cell. So this cell is known as the absorptive cell or the enterochine cell. So few names for these cells: epithelial cell of the mucosa, or the absorptive cell, or the enterochine cell of the small intestine. And here, this is the interstitial space, and here there is a blood capillary. One more figure I am showing you for this. For example, in this figure, here this is the pancreas. This one is the main pancreatic duct, combines with common bile duct and forms the ampulla, and ampulla opens into the duodenum. Then this is the accessory pancreatic duct, and it also opens into duodenum. So these two main and the accessory duct from the pancreas, they are pouring the pancreatic juice into the duodenum. Now in the pancreatic juice, the enzyme pancreatic amylase is there, and this enzyme is involved in the digestion of the carbohydrates in the small intestine. Now here in this figure, if you observe this figure closely, so this is the lumen. This space this space is the lumen of small intestine, and we know that this lumen receives chyme from the stomach. So once the process of digestion is completed in the stomach, the chyme enters into the duodenum. So now the process of digestion will start is about to start in the small intestine. So the process of digestion is taking place in the mouth with salivary amylase and in small intestine with the help of pancreatic amylase. So these two amylases are the main enzyme those are involved for the digestion of carbohydrate. Now here the cells, this cell, all this cell, this cells and this cell, so you can see that these cells are, they are arranged around the lumen of small intestine, and this cell are the absorptive cell or the enterocrine cell, and on the top, on the surface, they are having the microvilli. Okay, so the microvilli are there, and this microvilli together they form the breast border. For example, the same cell, this cell or this cell, so these cells are shown over here. Okay, these are the absorptive or the enterocyte cell, and here you see on the top finger like projections are present. They are very tiny one, and these projections are known as the microvilli, and many microvilli together they appear like a bristle of the brush. Their appearance of the microvilli, the appearance of the microvilli is look like the bristle of brush. So this border of microvilli, this border is known as the brush border. Now for the digestion of carbohydrate, on this border, on the brush border, enzymes are located, enzymes are present for the digestion of carbohydrates. For example, over here again in this figure, this is the lumen, this is the lumen of intestine. Then this cell are the enterocyte cell or the absorptive cell, and this one is the brush border, and these are the microvilli, and together they are forming the brush border. And on the brush border there are enzymes are present, and these enzymes are involved in the process of digestion of carbohydrate. And here, as you can see, the microvilli, the name is written. Now, so you need to understand, this is the lumen. Lumen is surrounded by the absorptive cell, enterocyte of the small intestine, and then after the cell there is a space. Okay, so this is the space, and then this space, after this space the blood capillary is present. So this is the blood capillary. So once the carbohydrates are digested, you see the starch, then disaccharide. So once they are digested, let's say the glucose is formed. So the glucose will enter into this cell. That is the process of absorption, and then the glucose will leave this cell. There is a mechanism, and then it will enter into the bloodstream or the blood of the capillary. So now we are moving back to the main figure. So this is the setup. Now you have the idea what this setup is, and here after this interstitial space, here there is a blood capillary. Now first we are discussing the digestion of starch. This digestion, either it is taking place in the mouth, in the buccal cavity or the oral cavity with the salivary amylase, or the digestion of starch, it is occurring in the lumen of small intestine with the help of pancreatic amylase. Now we know the structure of starch. In the structure of starch, amylose is there, and in the structure of starch, amylopectin is there. Amylose is in the linear form of glucose molecule. Amylose is having a linear chain of glucose. So these are all glucose molecules, and between the glucose molecule, this bond are the alpha 1,4 linkage. Okay, and this one is the amylopectin. Amylopectin is in the branch chain form of glucose molecule. So these are all glucose molecule. Now between the glucose molecule, alpha 1,4 linkage is there, this bond, alpha 1,4 linkage. All these bonds are alpha 1,4 linkage. Now this bond, see, this is one glucose chain, this one is one glucose chain. So at the terminal part here, this bond, okay, where the two glucose chain are joining, at that point this bond is the alpha 1,6 linkage bond. And this bond, which is very close to alpha, this is alpha 1,6 bond, and this is alpha 1,4 linkage, and this one is also alpha 1,4 linkage. So this linkage is, they are very near to alpha 1,6. So this one and this one, this bond is known as the adjacent alpha 1,4 linkage. And this bond is very far away, so this bond is known as terminal alpha 1,4 linkage. Okay, now alpha amylase, in the mouth salivary alpha amylase, in the small intestine pancreatic alpha amylase. Now how this amylase is acting, how this amylase act on amylose, and how this amylase act on amylopectin. How this amylase is digesting the amylose, how this amylase is digesting the amylopectin. That is the question. See, this amylase, and this is the amylose. Now in the amylose, these all are the 1,4 bond. What happens? This amylase enzyme can break this bond. So the bond which is breakable are shown with the dotted line. So this bond, amylase can break, this bond, amylase can break. So let's see, this alpha amylase is breaking this bond. So what will happen? These three glucose molecule are going to release, and they are having alpha 1,4 linkage. So together these three glucose molecule, they are known as maltotriose. Now alpha amylase can break this bond, So these two glucose molecule together they are going to release, and this combined two glucose molecule with the alpha 1,4 linkage, this molecule is the maltose. But amylase cannot break this bond, that is the terminal alpha 1,4 linkage, cannot be cut by amylase. Now over here, now which are the bond that the in the structure of amylopectin where the alpha amylase can break the bond? So the bond which are breakable, they are shown by the dotted line. So these are the breakable, and this dark brownish yellow color bond, they are non-breakable, they cannot be split. Okay, so in the structure of amylopectin, you see adjacent alpha 1,4 linkage is not breakable, then alpha 1,6 bond cannot be broken by amylase, then adjacent alpha 1,4 linkage cannot be broken down by alpha amylase, then the terminal alpha 1,4 linkage cannot be split by alpha amylase. So let's say the alpha amylase is splitting this bond and alpha amylase is splitting this bond. So all this glucose molecule which are having the adjacent 1,4 bond, then the alpha 1,6 linkage and adjacent 1,4 and the alpha 1,4. So this part is going to remove, okay, from the amylopectin by the action of alpha amylase. So we can say the alpha limit dextrin is released from the amylopectin by the action of alpha amylase. Then alpha amylase can act over here. So this two glucose molecule, that means the maltose can be released. Okay, then if I draw the third, so if we break this bond, so this three glucose molecule, that means maltotriose is going to release. So in other words, by the action of alpha amylase from the amylopectin, alpha limit dextrin is released, maltotriose is released, and maltose is released. So this is how the amylose and amylopectin they are digesting. This is how the amylase is acting on amylose and amylopectin, and this is how the maltotriose, maltose, and alpha limit dextrins are formed. So all these details that I have explained you is written over here. You can read this, all the information, and here you see as a result the product of alpha amylase action, a linear glucose oligomer maltotriose, that is the trimer of glucose, maltose a dimer of glucose, and alpha limit dextrin which is having alpha 1,6 branching linkage, they are formed by the digestion of amylose and amylopectin by the action of salivary or pancreatic amylase. So you need to read this, and this is the figure. So you observe this figure and you will understand what is happening. Now here, so now we are having alpha dextrine, maltotriose and maltose. So these three need to be further digested. So for that, here this one is the breast border, this one is the breast border, this entire. Then here you see this is this entire part is the lumen of small intestine, this is the breast border, and this part is the cytoplasm of the enterocyte cell or the absorptive cell. Now first we are discussing the digestion of lactose. Now what is lactose? Lactose is disaccharide made up of glucose plus galactose. From where we are getting this lactose? So when we are drinking the milk, we get, so from the milk we get the lactose. Now for the digestion of lactose in the breast border, there is an enzyme is present. So this enzyme, which is responsible for the digestion of lactose, which is present on the surface of breast border, this enzyme is known as lactase. And lactase enzyme splits the bond between glucose and galactose. So here you see what is happening. So this is the active site of the lactase enzyme, and this lactose is bound to active site of the enzyme, and this bond is cleaved off. So now what happens? The galactose and glucose are released by the action of this breast border enzyme lactase. So now where this glucose and galactose are going? So this one galactose, this is glucose. Now they are transported inside the cytoplasm of the intestinal epithelial cell or the absorptive cell or the enterocyte in the cytoplasm. But how they are reaching over there? So here you see this is the SGLT1, and this SGLT1 is Na plus, that is sodium ion Na plus coupled transporter. And what this SGLT1 transporter is doing, so it is transporting glucose and galactose inside the cytoplasm. And what is the mechanism how it is transporting this glucose galactose in the cytoplasm? That process we will learn in the absorption of carbohydrate topic. Okay, so in the next lecture we will learn that thing. So this is how the lactose is digested. Now on the breast border another enzyme is there. So this enzyme, this blue color enzyme, okay, so this enzyme is known as, the name of this enzyme is maltase, and maltase is also known as glucoamylase. Now this glucoamylase or the maltase, it can digest maltotriose or the maltose. And what is the mechanism how it is digesting this maltotriose and maltose. So you see maltase, this one, this is the active site, and this maltotriose attaches to the active site of the maltase. So what is happening? One glucose molecule is released. So now we are having two glucose molecules joined together. So that means this is the maltose, and one lonely glucose is formed. Then this side of the enzyme, this is active site, this is the maltose joined on this active side. So what is happening? Maltose is split into glucose plus glucose, and from this maltotriose we know glucose and maltose is formed. So this maltose will reach over here, will attach to this active site, and again this maltose will be broken down into glucose and glucose. So now both this glucose, they are transported into the cytoplasm of this small intestinal epithelial cell by SGLT1 Na plus coupled transporter, and this transporter is transporting the glucose molecule in the cytoplasm of the enterocyte cell. Now in the breast border there is a third enzyme, and this third enzyme, the name of this third enzyme is sucrase isomaltase. And you see over here it is written that sucrase isomaltase is actually two enzymes. So it is not one enzyme, it is two enzymes. So see here the sucrase moiety is shown, and here the isomaltase moiety is shown. So now what is happening? So which are the molecules are going to digested by this enzyme sucrose isomaltase? So here the sucrose is shown, then this one is the maltose is shown, then the maltotriose is shown. Okay, so here it is written the sucrase moiety of the enzyme splits sucrose as well as maltose and maltotriose. Now we know the structure, in the structure of sucrose there is this one is the glucose molecule, and this one is the fructose molecule. Maltose, glucose and glucose, and in maltotriose G and G, glucose and glucose. So these three are the glucose. So how this enzyme is going to digest? You see what is happening. This sucrose, where this sucrose is going, so it is attaches to the active site of the sucrase. So this bond is going to cleave off. So we are having glucose and fructose. This glucose and fructose, they are transported by transporter. The name of this transporter, though, that is the fructose transporter GLUT5. So this is how the fructose is transported. Then the isomaltase. Now maltose, when maltose goes over here active site, so this bond is going to cleave off, and we will have glucose and glucose. If maltotriose joins over here, okay, or here this place maltotriose join, then we will have glucose and glucose. Okay, so this is how the sucrase is acting. Then this part, this is the isomaltase. or isomaltase. How this isomaltase is acting? See, first we are sending the alpha limit dextrin. We are sending alpha limit dextrin to the active site of the isomaltose. So here you see on the active site of the isomaltase, this alpha limit dextrin is attached. And how this enzyme is acting? So it is breaking the bond, okay, and once the bond glycosidic bond is broken down, so alpha limit dextrin is converted into, you see, maltotriose and maltose. Now what happens to this maltotriose and maltose? So once the maltotriose and maltose are formed, again they are going back to the active site of the isomaltase enzyme. Malto, maltotriose is going for the digestion and maltose is going for the digestion. So when the bond are going to cleave off, when the bond are the broken down, so from the digestion of maltotriose and maltose, we are getting the glucose molecule, and this glucose is transported in the cytoplasm of small intestine epithelial cell by SGLT1 transporter. Okay, so this is how the entire digestion process of carbohydrate is taking place in the mouth and in the lumen of the small intestine. Now I am showing you the entire concept map. Okay, this way it is, this way, this is the half part of the figure, then this way, and all the information you need to go through, this very important information for you. And actually this is the single chart or single concept map or flow chart, but this single concept map is covering both the topics, that is the digestion as well as absorption. So if you want, with this figure you can, by your own, you can prepare the absorption of monosaccharide topic as well. Now I would like to show you all the figures before I leave. So this is what is this figure. See, this is the general mechanism of digestion and absorption, the figure. So where the process of digestion taking place, which are the molecules, protein, then carbohydrate, lipid, how they are digested and how they are entering in the epithelial cell, and then how the nutrients are reaching to the interstitial space. So you can have the brief idea about the digestion and absorption process with this concept map. Then notes, study notes for this. I have already delivered the lecture on digestion of carbohydrate in mouth. Then this way, then this, digestion of carbohydrate in stomach, then digestion of carbohydrate in small intestine, brush border enzyme of small intestine, then this, then this. this view. Now here some good information point wise they have given. So with this theory you can go through all the points. 1 2 3 4 and this figure. This is the information regarding the brush border. And this figure. This figure. So with this we have completed the discussion on carbohydrate digestion. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.